Hello, welcome to Lecture 2. This morning we're going to consider the electrical activity of the heart in two parts. I advise that you review the material in Levick's chapters 3 and 4. And here are your learning outcomes for at least the initial part of week 1. I know it's a lot, but we are going to work through each one of these and you're going to be in good shape at the end of the week. So if we were to monitor our pulse, in other words, palpate our pulse, what exactly does that mean? Where do the pulsations that we feel on the surface of the skin come from? Well, the pulse reflects the pressure wave generated by the rapid ejection of blood into the arterial vasculature. So the pulsations do not come from the veins. No, the veins are arranged more superficially, closer to the skin. But the venous pressures are simply too low, only 5 or 10 millimeters mercury, to be felt on the surface of the skin. So the pulsations are best felt above the arteries, not the veins. And if we are palpating the wrist nearest the thumb, on the thumb side, what we are feeling are the arterial pulse from the radial artery. If we're palpating the wrist on the little finger side, we are palpating the ulnar arterial pressure wave. Now, if we were to measure our pulse right now, we'd find a heart rate of 60 to 75 beats per minute. And if we were to monitor our heart rate over the course of a day, we'd find that this rate varies twofold with our daily activities. Posture, exercise, anxiety, general health, the number of espressos that we've taken after lunch, this all influences our heart rate. Heart rate even fluctuates from moment to moment, for example, with breathing. This is called respiratory sinus arrhythmia, and it's perfectly normal. Heart rate normally decreases during expiration and then increases during inspiration. Respiratory sinus arrhythmia, we'll talk about that more later in the week. So what triggers the heartbeat and what determines its rate? Does the brain control the heartbeat? Well, it's true that the heart is innervated by both branches of the autonomic nervous system, parasympathetic and sympathetic, and these nerves do influence rate of the heartbeat. But in fact, the heart can function efficiently without any nervous input. Consider the isolated cardiac myocyte sitting in a dish of fluid. It can spontaneously contract with an inherent rhythm all its own. This one happens to be dead, but this one and this one are alive, and notice that they are spontaneously contracting with an inherent rhythm all on their own. Or consider a patient that has received a heart transplant. Their new heart is completely denervated. The surgeon does not reattach the vagus and the stellate nerves to the heart, to the new heart. But in fact, these patients can function quite well, and the transplanted heart can even increase its output in response to exercise. Imagine that. We're going to find out how that occurs later on. So this demonstrates that the heartbeat is initiated from within the muscle cells themselves. No external signals are required. So how are these contractions of the 3 billion individual cardiac myocytes, how are they coordinated to give a contraction that is coordinated in space and time? Well, in order to function efficiently as a pump, 5 conditions actually must be met. So for efficient pumping, contractions must be regular. They have to occur at regular intervals. They must not be arrhythmic. And the contractions must be coordinated not only in time but in space. For example, the atria must contract before the ventricle, and the ventricle must contract from its apex, squeezing the blood upward toward the outlet port. The contractions must be sufficiently forceful. A failing heart has a weak contraction and therefore a weak output. And the valves, when closed, must not leak. They must not be insufficient or regurgitant. And finally, the valves, when open, must be fully open. They must not be narrowed or stenotic. So today, we're going to consider the 2 requirements. The contractions must be initiated at appropriate intervals, and they must be coordinated in space and time. The heart is made up of about 3 billion slender muscle cells. A single fiber is called a cardiac myocyte or a cardiomyocyte. And these rod-shaped cells branch and connect with one another end to end, and they join at structures called intercalated discs. This is where finger-like projections of each cell interdigitate. And at these interdigitating projections, we find channels in the cell membrane called gap junctions, button-like clusters of membrane proteins called gap junctions. And these form large pores between adjacent cells. And the gap junctions allow ions and small molecules, some as large as ATP, to pass freely from one cell to the next. So gap junctions provide a pathway for electrical current in the form of ions to flow from cell to cell within the cytosol. And since all muscle fibers of the heart are electrically coupled, an action potential in one cell will be propagated through the myocardium as a wave of excitation. So the myocardium, in effect, acts as a functional syncytium. The word syncytium simply means a single giant cell, and this is with respect to electrical excitation. So the heart has 3 basic types of muscle cells, and each is specialized to perform a different function. Most of the cells, 99%, are contractile cells. They make up the working myocardium, and we find at specialized locations highly specialized muscle cells that form the SA node, the sinoatrial node, and the atrioventricular node, the AV node. And at regular intervals, these so-called nodal cells spontaneously generate an action potential. They're kind of like little spark plugs. And they function as pacemakers that trigger the heartbeat at regular intervals. The third type of muscle cell is called a Purkinje fiber. And it forms narrow tracks, kind of like little wires, that distribute electrical excitation throughout the heart rapidly. Now here we see a cross-section of a sheep's heart, and it very nicely shows a stained Purkinje fiber network, the wiring network of the ventricle. Here's another image, histological image, which shows the Purkinje fibers located just below the endocardium or inner surface of the heart, the ventricle. Here's the endocardial surface. Just below, in the subendocardial connective tissue, we find the Purkinje fibers. Note that the Purkinje fibers are very large. They are much, much larger than a cardiac myocyte. And because of this large diameter, and because they have high density of sodium channels, they conduct excitation very rapidly. The action potentials spread along the Purkinje fibers extremely rapidly. Notice that the Purkinje fibers have a few myofibrils, but very, very few. They're highly specialized, and they lack T-tubules. So it's important to recognize that all 3 of these muscle cells are excitable. They can all transmit action potentials. All 3 are capable of so-called automaticity. That is, they can all initiate action potentials at regular intervals. And they are all muscle cells with varying degrees of specialized differentiation. Okay, the internal wiring system of the heart, the conduction system, is shown in this illustration as colored tracks. The impulse begins in the SA node, the sinoatrial node. The SA node is about the size and shape of a grain of rice. It's located in the groove between the superior vena cava and the right atrium. And it's the normal spark plug of the heart. The SA node functions as the normal pacemaker of the heart. And about 70 times each minute, the SA node emits an action potential that spreads through the wall of the atrium. The impulse may also travel through specialized conduction tracts known as internodal tracts, shown here in green. But the existence and importance of these tracts is controversial in humans. Let's just assume that they exist. They do exist, but The impulse can also travel from atrial myocyte to atrial myocyte. And the impulse then arrives at the AV node, situated right here. And the AV node performs three important functions. First, the AV node relays the impulse from the atria to the ventricles. Recall that the AV node is normally the only electrical connection between the atria and the ventricles, because the atria are electrically insulated from the ventricles by the so-called fibrous skeleton. So excitation can only pass normally through the AV node. Now there is one exception in a congenital disease called Wolff-Parkinson-White syndrome, WPW syndrome. And this is where there is an abnormal accessory path, typically here or here, that between the atria and ventricles, and this can cause a premature and uncoordinated excitation of the ventricles. We'll see that in a little bit. Okay, in addition to transmitting the excitation across the fibrous skeleton, the AV node delays the impulse before passing it on to the ventricles. Now think about this. This delay is absolutely crucial because that allows the atria enough time to help fill the ventricles before they're excited. Remember, the atria serve as small booster pumps to the ventricles, helping to fill it in the very last phase of ventricular filling. And this delay allows the atria to contract and top off the ventricles at the very end of filling. So the AV node serves as a spatial and temporal bottleneck for the excitation of the ventricles, a spatial and temporal bottleneck for excitation passing into the ventricles. The AV node can also function as a backup pacemaker if the SA node should fail, or if there is a conduction block between the SA node and the AV node. Next, the AV node passes the impulse onto a thick tract of Purkinje cells called the bundle of His, located at the top of the septum. And these fibers, remember, Purkinje fibers have an extremely rapid conduction velocity. These fibers conduct the impulse very quickly along the septum in two branches, the left and the right bundle branch, and these branches then repeatedly branch further to form the Purkinje system, which projects to all parts of the ventricle. Now, the Purkinje fibers ramify into the endocardium. Along the surface of the endocardium, remember, the Purkinje fibers run along the surface of the endocardium and then project inward towards the epicardium. So if the muscle fibers of the heart are electrically coupled by gap junctions, why does the heart need its own wiring system? Well, first, the conduction system allows the impulse to travel fast. Propagation from cell to cell would just be too slow. And second, the conduction system guides the impulse, channeling it in the proper direction at the proper rate so that the excitation is delivered to specific regions of the heart at specific times. And this allows for coordinated, efficient pumping. To understand how excitation spreads through the heart, we have to consider certain special features of the cardiac action potential. Suppose that we use a slender glass microelectrode to record the membrane potential in a single cardiac myocyte in the ventricle. Now it would, after we impale the cell membrane with the electrode, we would record a resting potential of about minus 70 to minus 90 millivolts. But at regular intervals, we would see a wave of depolarization move past our electrode, and this would result in an action potential with an odd shape, like this. Now, suppose we took our microelectrode and impaled muscle fibers in all different regions of the heart. If we inspect recordings from hundreds of locations, we would find basically just two types of action potentials. Fast response action potentials, these are found in all working myocardial cells. So contractile cells and Purkinje fibers would show these so-called fast response action potential. And the only other region of the heart to display a different profile of the action potential would be in pacemaker cells, the AV node and the SA node. Now, looking at the differences in the action potential waveform, you might be able to predict why nodal cells function as pacemakers. As we just mentioned, their resting potential never really rests. There is a depolarizing drift until the action potential is triggered. And you might be able to predict why the nodal tissues, the nodal cells, conduct impulses very slowly. So this is a very informative plot. It shows the conduction velocity in meters per second through the various components of the conduction system against time. So notice that the SA node and the AV node have conduction velocities that are 50 to 70 times slower than the working myocardium or the Purkinje fibers, which are sort of the king of conduction velocity at 3 meters per second. Think about that, 3 meters per second versus the SA node at 0.01 meter per second. So the impulse is initiated in the SA node, shown here, and then it propagates in the following manner through the conduction system very, very rapidly. This is the part that is important, and that is AV nodal delay, this delay right here. And it's all because of the slow conduction velocity through that AV node. So how does excitation spread through the heart? Well, in this illustration, the red regions of the heart are depolarized and the blue regions remain hyperpolarized. So remember, excitation starts in the SA node and spreads through the atria. After the atria are completely depolarized, the excitation is passed by the AV node across the fibrous skeleton into the ventricles. And the first part of the ventricles to depolarize is the left portion of the septum. The apex of the septum is then depolarized. The right part of the ventricle depolarizes predominantly first, and then the very last portion of the ventricle to depolarize is the epicardium of the left ventricle. Here's a situation where the atria have been repolarized fully and the ventricles are depolarized fully. And then the process of ventricular repolarization begins, and it's a little strange because it begins on the left ventricle first from the epicardium toward the endocardium. We're going to talk about this in much greater detail when we consider the electrocardiogram, because this is entirely, these electrical vectors that change in space and time are the origin of the waveforms of the electrocardiogram. So let's take another look at that little animation of the heart and the conduction system, and let's point out a few important components. First, note that excitation normally begins and spreads in a unidirectional manner from the SA node, and that local excitation initiates local muscle contraction. Excitation then passes from the atria to the ventricles only via the AV node, and it passes very slowly and delayed momentarily. All other pathways of excitation are extinguished by the fibrous skeleton. So this is normally, the AV node is normally the only pathway for excitation of the ventricles. Then excitation is rapidly distributed throughout the ventricles by the Purkinje network, starting mainly in the apex of the heart. Why might the excitation of the apex first be important? Think about that. And then as the ventricles are contracting and ejecting their contents, the atria are simultaneously refilling with venous blood. Okay, that concludes part 1 of today's session. Grab a cup of coffee and take a little break, and we'll return momentarily with part 2 video. Thank you.